Okay. Let me know. Can you see the screen? What can you see?
Yes, ma'am.
Ah, so what we are going to start is the third step. After Krebs cycle, the next and the last step which we want to learn, we have to learn is ETS, that is electron transport chain or electron transfer system, ETC or ETS, that is called as. Okay. Now with the help of this diagram, I will explain you. Now that day I told you that in Krebs cycle as well as in the uh, glycolysis, which type of energy is produced? That is ATP. You studied ATP is produced as well as NADH2, FADH2, right? Now, how this ATP is generated or how this energy is produced during the transfer of these electrons, that we will see. Now, two first is the substrate level electron transport directly from, that is the formation of ATP. So you know another name for this ETS is also called as oxidative or oxidation reaction or oxidative phosphorylation. How it is called, I will explain the system and then I will tell you. So first of all, let me explain this. The carriers which are present, they are electron carriers. Okay, as I told you that these F1 particles or the oxysomes in the mitochondrial inner membrane, they are playing very important role. So wherever NADH2 and FADH2 are produced during glycolysis, connecting link reaction, that is acetylation, and Krebs cycle, they are oxidized with the help of various electron transfer or electron carriers and enzymes. Now what are these electron carriers and enzymes? See, let us see. These carriers and enzymes are arranged on the inner mitochondrial membrane in the form of various complexes as complex 1, 2, 3. This is 4 actually. This is printing mistake, I think, and 5. So NADH plus H, that is NADH2, is oxidized by NADH dehydrogenase, that is complex 1, and its electrons are transferred to ubiquinone, that is coenzyme Q. This is represented as COQ, present on inner membrane of mitochondria. And reduced ubiquinone is called as ubiquinol. So FADH2 and the next another substrate, that is the another enzyme, that is FADH2 coenzyme, is oxidized by complex 2, that is succinate dehydrogenase, and these electrons are also transferred to coenzyme Q. During oxidation of NADH2 and FADH2, electrons and protons are released, but only electrons are carried forward, whereas protons are released into outer chamber of mitochondria. Outer chamber of mitochondria, they are released. What is present over there, how they are carried, that I just mentioned while we were revising. Okay, now see how this electron transport system works. Now this is it is mentioned here. This is the mitochondrial matrix, then inner membrane, mitochondrial membrane, and this is the space in between the inner and outer membrane. Okay, so this is the mitochondrial matrix where this NADH2 is present. Then that gets, in presence of NADH dehydrogenase, converted into NAD, that is oxidation reduction reactions are taking place. NADH2 and FADH2, they are oxidized, isn't it? They are oxidized by the enzyme called NADH dehydrogenase, that is complex 1. Okay, so what happens then? NAD is produced, and NAD then gets converted first into FMN, flamine mononucleotide. So then FES, which is the, again, you know, these are ion containing electron carriers and enzymes, coenzymes present in the mitochondrial matrix, you know. So this NADH2 first gets oxidized in presence of enzyme NADH dehydrogenase and gets converted into FMN. Now during this process, electrons are released and some amount of energy is released, that is stored in the form of ATP first. This is the first step where ATP is released. Then through FES, these are accepted by ubiquinone. Okay, that is coenzyme Q. Ubiquinone or coenzyme Q. This is what it is shown. And then these electrons are carried to cytochrome B first. See, this is the ubiquinone. Ubiquinone. From there, these electrons are accepted by the next complex, that is called as cytochrome complex 3, where this first is accepted by B, that is cytochrome B, and then cytochrome C, that is, this is called as BC complex, that is cytochrome BC1 complex, and then it is accepted by C, cytochrome C. Here it is shown here as B. This is through then accepted by cytochrome C1 and then to cytochrome C. So this is not straightway it is shown here, but I am talking about only the one coenzyme, only one complex, that is NADH2. How from NADH2 these electrons are transferred? I'll repeat again. NADH2 gets oxidized to NAD in presence of NADH dehydrogenase. Okay, so this is the first complex. Then these electrons are accepted by FMN, flavine mononucleotide, where this energy which is released during this transfer is stored in the form of ATP. That means from ADP and IP, ATP is produced. This is the first ATP produced here. Then through FES, these electrons are carried to coenzyme Q. This is also called as ubiquinone. Then when it is oxidized, this is called as ubiquinol. Just now I read, right? So ubiquinol, from there these electrons are carried to the next complex, that is complex 3, where cytochrome BC1 complex, this is called as. That means these electrons first are accepted by B, then to C1, and then this, during this transfer again, second ATP is generated. That means this is the first ATP generation when we are using NADH2. So this is the first ATP generated. Here it is not shown, but remember that this is the first. Then through ubiquinone to ubiquinol, these electrons are transferred to complex 3, that is called as cytochrome BC1 complex 3. So first accepted by cytochrome B and then to cytochrome C1, and then during this transfer of electron from this complex, the whatever amount of energy during this transfer of electrons, the amount of energy which is released is stored in the form of ATP. So here is the second ATP for synthesis taking place. So second ATP is generated or formed or synthesized. Then from BC1 to C, these electrons are carried to cytochrome C. This is a small protein, iron containing protein. These are all the iron containing, some are iron containing proteins. These are all different types of proteins. Okay. So then from cytochrome C, these are accepted to the cytochrome C. From cytochrome C, these are accepted by the next complex, that is complex 4, that is called as cytochrome A3 complex. That is first accepted by A and then by cytochrome A3. So this is called as next fourth complex, that is the cytochrome C to A3. Then at the end, from this complex, the electrons are carried to the molecular oxygen, and then this oxygen which is present here, this oxygen accepts this electron. At the end, this is the oxygen is the product, or oxygen is the last one which accepts the electron. So this is called as, where the reaction ends, this is called as terminal oxidation. And this oxygen, by accepting electrons becomes negatively charged, and during that process whatever these hydrogen ions are released, that means protons, at the same time these electrons are carried. I just told you before this that electrons are carried from these electron carriers from through F1 particles, whereas you know that the base which is called F0, these protons are transmitted, and there these hydrogen, that is that protons, these are accepted by oxygen to form a water molecule that is called as metabolic water. So this is the last step, where at the fourth step here, when it is accepted, when these electrons are accepted by oxygen, there again some amount of energy is released at the end, and this is the third ATP produced when we are using, or there is oxidation of NADH2. Okay, so oxidation is either addition of oxygen or removal of hydrogen. So as it is removal of hydrogen, the enzyme, the names are also given as per the reaction and the type of substrate they use. So the enzyme is NADH dehydrogenase. Okay, I explained you with the help of only taking oxidation of NADH2. So how many times the ATP is generated? This is the first step where NAD gets is accepted by FMN. So NADH to NAD oxidation. Then it is accepted by FMN. There is the first step where first ATP is generated. Then second step from BC1 to C. This is the step where from BC1 complex to C, second ATP is generated. Okay, this is the second ATP generated. And the last one, when terminal oxidation takes place, so from A3, when it is accepted by oxygen, then this is the third ATP generation. So from one molecule of NADH2, how many ATP are generated? 3 ATP are generated. This is the first step where it is taking place during the conversion of NADH2 to NAD and then to FMN. Second is BC1 to C, transfer of electrons from BC1 complex to C. Second ATP is generated, and the third one, it is generated at the end, that is terminal oxidation, when it takes place when oxygen accepts the electron. So oxygen is the last one to accept the electrons. So at this stage again the third ATP is generated. So from one NADH2, 3 ATPs are generated, and this at it ends the reaction. So this is called, and it is taking place, oxidation is taking place, so this is called as terminal oxidation. Whereas by using these protons, when they join with this oxygen, molecular oxygen, there is formation of metabolic water, and here is the oxidation reaction taking place. So this is what, and at the same time here there is phosphorylation taking place. Phosphorylation is what? Addition of phosphates. So ADP plus IP giving rise to formation of or synthesis of ATP. So that is what called as oxidative phosphorylation. So the names for ETS is called is, first is electron transport system or ETC, that means electron transfer chain, transport chain, or that is what. And the third one, terminal oxidation, the same process, and it is terminated by oxidation. At the same time phosphorylation is taking place, that means there is generation of ATP. So that's why this is called as oxidative phosphorylation. So every time you remember all these names, ETS is nothing but not only electron transport chain or electron transfer system, but it is also called as terminal oxidation, because it, that last acceptor of electron is oxygen, where oxidation is taking place at the end, so terminal oxidation. And during that process water molecule is formed, that is called as metabolic water. And this oxidation gives is correlated with phosphorylation, that means there is synthesis. Phosphorylation is nothing but what? Addition of phosphates. So ATP is produced from ADP plus IP, that means phosphorylation is taking place, and that is oxidative phosphorylation. So this is also called as oxidative phosphorylation. I hope you have understood up to this. Now I will talk about the when FADH2 is used. Now here, this is the second substrate where FADH2 gets converted into FAD, oxidized to FAD in presence of enzyme succinate and succinate dehydrogenase, or this is what you succinate dehydrogenase react is the enzyme where it FADH2 is oxidized by complex 2, that is succinate dehydrogenase. And during that transfer, how many ATPs are generated? Here you can see that here also when FAD is oxidized first and then transferred, that the electrons are carried, transferred, these are accepted by cytochrome C via this conversion, first to ubiquinol and then to cytochrome C. So here also the same thing, first acceptor is, see here, FAD, first acceptor is ubiquinol, there it is FMN. So during that transfer, this is the first ATP in NADH2. But here FMN is the first one conversion. But here directly this FAD first accept, these electrons are accepted by ubiquinone, and then through ubiquinone again to BCB and then C1. So during this is the first step where ATP is generated here. So FAD is oxidized, and then during that transfer, the electrons which are released, they are accepted by ubiquinone. Then ubiquinol is the name when it is oxidized. These are accepted by B and C1. So this is called as BC1 complex. And then from BC1 to C, so this is the first generation of ATP here. From BC1 to C, this is the first ATP generated when the substrate, that is the FAD, is used. Okay. And then rest of the part is same. Then from C, they are carried to A1 or A and then A3. And then from A3, oxygen is the last one to accept these electrons and forms this molecular oxygen, then combines with the protons. That means you can see here when these are all carried by F1 particles, and these are the F0 bases, that is the F0 bases which are providing the space for transport or release of these protons, and these protons are then accepted by these molecular oxygen to form a metabolic water. This is what when NADH2, this is the first ATP generated, additional one, and when FADH2 is there, the rest of the things from BC1 to C, first ATP for this, and then next is from A3, this is what A to A3, this is what the last one. So rest of the things are same. When NADH2 is oxidized, this is the first step where the first ATP is generated. Rest two are common in case of FADH2 and NADH2, same. Okay, so remember this, that in this way there is ATP synthesis taking place, that means phosphorylation taking place. So this is also called as oxidative phosphorylation. Now before we come to this step, I just want to show you few slides, and then I will show you the total production or net benefit or the total synthesis of ATP generated during this entire cycles, that is called as balance sheet for ATP by aerobic oxidation or aerobic respiration of one glucose molecule. For that, you keep your textbook open for glycolysis as well as, then when I will tell you, you will be answering me. Okay, I will show you few slides first, and then we will can you see this now? What is this? Just let me know. Can you see?
Electron transport system and oxidative phosphorylation.
Have you understood the meaning of electron transport system and oxidative phosphorylation? Electrons are transported with electron carriers, so electron transport system, and as it is taking place phosphorylation, that is in presence of oxygen, oxidation is taking place, oxidation reduction reaction, that is total entire one. So oxidative phosphorylation, phosphorylation is what? Addition of phosphates. So now if you can understand this diagram now. Okay, This is the one and the same, but in a different manner it is shown, because here these are the complexes. This is complex 1, complex 2, complex 3, then complex 4, and at the end this is ATP synthesis, where all these are proteins. This is the mitochondrial matrix. This is the head part, you can say, and this is the base part. These are all electron carriers, where this is the first complex when NADH2, where NADH2 gets converted into oxidized to NAD and then accepted by FMN. So this is where electrons are carried, and then here are the protons. They are released from base to base. They are transported. They are released, and in the this mitochondrial membrane. Okay, and then this is the Q. That means the coenzyme Q, or this is also called as ubiquinone. Okay, and these are all proteins. And then so that's why this is shown that these are different types of proteins, iron containing proteins. Most of these are iron containing proteins. Then from ubiquinone, cytochrome C. Then cytochrome C, not directly cytochrome C. This is the complex. What is it? BC1 and then C. Then to fourth via A3, A1, A3. So when NADH2 is the substrate, when NADH2 is used, 3 times ATP is generated. When this is the first when it gets converted into FMN, and these are the electron carriers. So just synthesis of ATP is taking place here. So this is phosphorylation, and this is the end part where this is also considered to be fifth. This is considered to be fifth complex where ATP synthesis is taking place. Okay, just try to understand. Doesn't matter though you don't understand everything, but at least whatever you understand, you try listen carefully, and at the end we will be having discussion for your doubts. Okay. So series of proteins in the mitochondria helps transfer electron from NADH to oxygen. So electron transport system involves all these multi-protein complexes 1 to 5 and 2 mobile carriers, that is ubiquinone and cytochrome C. This is the same reaction I have taken here from the textbook. So these are the various levels of aerobic respiration. And when complex 1, that is dehydrogenase enzyme, is working, where electrons transfer from NADH to ubiquinone, and ubiquinone is located within the inner membrane, and it is mobile carrier between complex 1 and 3 and between 2 and 3. That means for NADH2 also and for FADH2 also, isn't it? So this is the second complex, that is the succinate dehydrogenase, where electrons are transferred from succinate to ubiquinone, that is FADH to ubiquinone. So that's why I said this is the second complex. And third is what? Where cytochrome C is a small protein attached to the outer membrane of outer surface of inner membrane, where cytochrome C, there is reduction of cytochrome C by transfer of electrons from ubiquinol via BC1 complex, if you remember. Okay, and the last one is AA3 complex, that is fourth complex, that is cytochrome C oxidase enzyme is used, where electrons are transferred and reduced from cytochrome C to molecular oxygen, where it contains 4 units, A3 and 2 copper containing proteins. Okay, so this is all about the transfer of electrons in the form of ETS. Mechanism of ATP synthesis is explained by chemoosmotic hypothesis. Now you don't stress on this word. This is a little bit different meaning it is giving. So I don't want to give you any or keep you in that confusion mind. So just let it be about that part. We will discuss as and when it comes. So this is what ATP synthase, which is used in this ETS during the synthesis of ATP from ADP. And as I told you that it contains 2 major components, F1, that is the base piece, and F1 is the head. So F1 is the peripheral membrane protein complex and contains site for synthesis of ATP from ADP and IG, that is inorganic phosphate. F0 is integral membrane protein complex that forms the channel through which protons cross the inner membrane. I showed you in the reaction itself, in the diagram itself. The passage of protons through the channel is coupled to the catalytic site of F1 components for the production of ATP. So for each ATP produced, 3 protons pass through F0 from inner membrane space to the matrix down the electrochemical proton gradient. If you, though you don't understand, doesn't matter. We will first discuss about this balance sheet, respiratory balance sheet. This is also called as. Now during this all the steps, how this ATP is synthesized, that we are going to discuss. So it is possible to make calculations of the net gain of ATP for every glucose molecule oxidized, but in reality this can remain only theoretical experience. So only theoretically we have to remember, okay, because every time it doesn't take place one and the same. It depends on the substrate which is used. Okay, so there is sequential orderly pathway functioning with one substrate forming the next, and with glycolysis, TCA cycle, and ATS pathway following one after another. The NADH synthesized in glycolysis is transferred into mitochondria and undergoes oxidative phosphorylation, which we discussed just now. So glucose is being respired, only glucose is being respired, no other alternative substrates are entering in the pathway at any of the intermediate stages. Now we will see how there is a difference. What is the difference between the fermentation and aerobic respiration? First, fermentation is partial oxidation of glucose molecule, whereas aerobic respiration means complete oxidation of glucose molecule. Okay, then net gain of ATP in fermentation is only 2, whereas in case of uh, aerobic respiration it is 38. Just make the change. This is little uh, change you have to make. This net gain of ATP is 38, not 36. Okay, this is what you have to make the change. NADH is oxidized to NAD slowly in case of fermentation, and it is vigorous process. Now how this balance sheet, now you open your glycolysis page, whatever the page is there, and you will be answering me. Okay, unmute yourself when I will be asking you. First thing, glycolysis. Keep that glycolysis page open. I will ask you. ATP produced by substrate level phosphorylation. Now, can you see the step where first ATP generated, that is 1,3 biphosphoglyceric acid to 3-phosphoglyceric acid? Tell me here. ATP is generated after the splitting. Do you can you see this on the page of glycolysis pathway? As there are 2 molecules, so there here it is 2 shown. So 2 ATP are generated, and the last step of glycolysis, phosphoenol pyruvic acid to pyruvic acid, PEP to pyruvate or pyruvic acid, PA. Last step of glycolysis, where this with the 2 molecules, there are 2 molecules, so there are 2 ATPs generated. So total how many? 4 ATPs are generated during this first step of glycolysis. Remember, open that page, then come to the same page again. You can see when glucose gets converted into glucose 6-phosphate, the first step where 1 ATP is utilized, and then the third step, I think, just keep the pathway open and see, fructose to fructose 6-phosphate to fructose 1,6 biphosphate. There also one utilization of ATP is there, right? I think first and the third step, just check. I don't have this in front of me. So out of 4, 2 are utilized. So what is the net gain? 2. So at the end of glycolysis, total ATP generated are 4, but 2 are utilized. So total net gain of this glycolysis is 2 ATP. Then next, from second step, that is acetylation step. Do you remember? Here 3P that pyruvic acid, or you can say 2 biphosphoglyceric acid. When from the same, that is glycolysis, here we will see that is that step where NADH2 is produced. two NAD molecules are there. So this step, 3-phosphoglyceric acid, 2 biphosphoglyceric acid, each worth of 3 ATP. There are two molecules. So 2 into 3. Here is one more change you can make. Here is the mistake where instead of 2, it should be 3. So 3 into 2, 6 ATPs are generated. Whereas ATP gain from glycolysis is in presence of oxygen is this is 6, and the previous one which we saw just now directly produced ATP 2. So 6 plus 2, how many? Total 8 ATP. So you remember, at the end of glycolysis, how many ATPs are generated? Directly produced ATPs 4, 2 utilized. So total 2, net gain of ATP 2. But from using NADH2, 2 NADH2 molecules are produced. So 1 is equivalent to 3 ATP. Just now we studied in ETS. So 3 into 2, it is 6. So 6 plus 2, it is 8 ATP. So at the end of glycolysis, net gain of ATP is 8 ATP. Now the next middle step, that is called as acetylation reaction. If you remember, pyruvic acid, when it gets converted into acetyl coenzyme A, NADH2 produce, there are two molecules. As there are two molecules of pyruvic acid, here two molecules of NADH2 are produced. And you know, one molecule of NADH2 is equivalent to 3 ATP. So 2 into 3, 6 ATPs are produced during this linked reaction, that is, which is considered to be connecting link between the glycolysis and Krebs cycle. This is also called as acetylation reaction or oxidative decarboxylation. Okay. Then remember, 8 at the end of glycolysis, 6 during the connecting link, that is acetylation from NADH2. Then the third now, Krebs cycle, directly produced ATP are when there is conversion of succinyl coenzyme A to succinic acid. That means first GTP is produced from GDP plus IP and then immediately converted into GTP. So two molecules are produced there. So 2 ATP are generated from 2 GTP during this conversion. Okay. Then next, when NADH2 is a substrate, when from NADH2, how many are produced? These are the 3 steps where isocitric acid gets converted to oxalosuccinic acid, alpha ketoglutaric acid gets converted to succinyl coenzyme A, and malic acid when gets converted into oxaloacetic acid. During all the 3 steps conversion, 2 molecules of NADH2 are produced every time. And so as one NADH2 is equivalent to 3 ATP, 2 into 3, 6, 6 into 3, 6, 6, 6, 3 steps. So how many total? 8, 6 3s are 18. Then FADH2, when it is considered, so from succinic acid to fumaric acid, 2 molecules of FADH2 are produced, and that one FADH2 is equivalent to 2 ATP. Just now we studied in ETS. So 2 into 2, 4. Okay. And ATP value of Krebs cycle is now 24. Do you remember how it is 24? 86 plus 6 plus 6, 18. 18 plus 4, how many? 18 plus this 6. This is again 2. That is 18 plus 2, 20. And 20 plus this 4, 24. So from FADH2, from NADH2, and from ATP directly 2 plus 18, 20 plus 4, 24. So from Krebs cycle, total ATP gain is 24. Now net gain of ATP in aerobic respiration is A plus B plus C. A is glycolysis, that is 8. B is acetylation, this is 6. 8 plus 6, 14. Am I right? Then 24. So total is 38. So A plus B plus C, 8 plus 6 plus 24. So that it becomes 24 plus 6, 30, and plus 8, 38. So note total net gain of ATP during aerobic respiration is what? 38 ATP. Just remember, the same thing is shown in your textbook in a tabular form. In this you can see glycolysis. During glycolysis through phosphorylation, 4 ATPs are generated but 2 are consumed. And from NADH2, two molecules of NADH2, that means total 6 ATP. So 6 plus 4, 10 plus 2 are consumed. So net gain is 8 at the end of glycolysis. Now pyruvate to acetyl coenzyme A, that means acetylation reaction directly from NADH2, two molecules of NADH2 are produced, which is equivalent to 6 ATP. So total is 6 at the end of acetylation reaction. And then from Krebs cycle, total gain of directly from phosphorylation to ATP are produced. From NADH2, 6 molecules, so 18. And from FADH2, 2 molecules, that means 4. So total is 22 plus 2, that is 24. So the total comes to here, total 42 utilized, so is 38. Net balance sheet, this is called as ATP, balance sheet of ATP by aerobic respiration or by oxidation of one glucose molecule. Okay, you understand this? Now just I will stop my presentation also, and then we will be having little discussion.